I'm Kayla Nolette.
I'm Leighton Knoll.
My name is Miguel Velez.
And I'm Olivia Olette.
And we also have another member, Victoria Holbert, who couldn't make it here today. And our experiment was the growth rate of Lactobacillus acidophilus in microgravity.
This science competition was opened up to 1,400 students at Montachusett. There were 27 different proposals submitted, and 3 were chosen out of the 27. And these were the growth rate of Lactobacillus acidophilus, HeLa cell growth in microgravity, and the acceleration of porphyridium reproduction in microgravity. And out of these 3, there was a final one, and that was the growth rate of Lactobacillus acidophilus.
I designed this mission patch for Montachusett to represent our experiment that got to fly in space. The shuttle represents the historic last flight of the shuttle Atlantis, which carried our experiment into space. The gear represents not only Montachusett's experiment that got to fly into space, but also the fact that we are the only vocational school that is participating.
So our proposed question, we wanted to know whether or not the bacterial Lactobacillus acidophilus would reproduce in greater numbers in space, in microgravity, or here on Earth. And there's a great importance to this question because Lactobacillus acidophilus is, like they said earlier, an acid, and what it does is it decomposes the enamel on the teeth. And after a time, if it's not removed, it can really start to do damage. So what we wanted to do was basically find out if this Lactobacillus acidophilus is going to increase in numbers in space. How are the astronauts going to actually protect themselves while they're in space from these kinds of things? And our hypothesis was that Lactobacillus acidophilus would increase in numbers in microgravity due to the fact that through past experiments there has been bacteria have been brought up to space, and a lot of times it's been a lot more active and reproduced in a lot greater numbers, so we figured maybe this would be the same way and do that same thing.
In our experiment, we had 3 variables, the independent variable, the dependent variable, and the constant variables. The independent variable tested was the reproduction rate of the Lactobacillus acidophilus in normal gravitational conditions. And the dependent variable tested was the reproduction rate of the Lactobacillus acidophilus in microgravity conditions. And then our constant variables were the volume of the capsules, the spore samples from the same vial, the nutrients used for the bacteria to consume, the time in which the bacteria reproduces, and the concentrations of the material. And the materials we used in our procedure was Lactobacillus acidophilus bacteriospores from Carolina Biological, and the 10% mass to volume of milk to tomato juice, and the 5% mass to volume of the yeast suspension prepared by Carolina Biological, the MDA type 1 experiment slot, the dental probe, the micropipette, and the cotton which held it all together. And for our procedure, what we did was we filled 3 capsules to capacity with the cotton and the Lactobacillus acidophilus spores, and then we took 3 more capsules that we filled to capacity with the tomato extract.
So to obtain our results, we had a variety of different tests that we used, and the first of these tests was an optical density test. And the way we did this was by basically taking both samples, the one from microgravity and the one from here on Earth, and we put them into this scanner that was called the tunable diode laser absorption spectroscopy scanner. And so what we did with that was basically, what that would measure is how dense the different samples were. So it uses a light to do this, and at the end of the experiment from the optical density scanner, we were able to see how dense each of the things were. And at the end of it, there was a significant difference after doing it a variety of times that the one in space was denser, indicating that there was more bacterial cells in that sample. Next, we did a lactic acid procedure. And basically what we did is lactic acid is a waste product from the bacteria after they eat their nutrient. And so what we did was basically we wanted to see how much lactic acid was in the samples. So first we prepared the samples, then we created a standard curve, and after that we added the reaction mix and finally measured the optical density once again. And so this right here is our lactic assay results. And right here we have on the left side is the nanometers, which is a light wave measurement. On the bottom is the nanomoles, which is the amount of lactic acid. And we did this so that we can compare all of our results back to a standard. And so right at the top right of the screen, you can see an equation. And that equation is what we plugged in our numbers to get our final results. And then this was our lactic acid results. We split our samples into 3 different wells, well 1, well 2, well 3. And this right here is the ground truth, and here we have the space truth. And this is well 1, well 2, and well 3 also. And so basically right here we're measuring the lactic acid within the different wells. And you can see how they're all practically very close in number. And then we got the average of them. This is the ground one here, and the average of the space one here. And we plugged those numbers back into our standard curve equation to finally get our number after we had to basically increase everything by tenfold due to the fact that we diluted the samples out in the beginning of the procedure. So after we did that, we got our final answers. Here, the ground truth was 2.7, and then we have 3.3 for this space experiment, which shows that there's a greater amount of lactic acid in the space experiment, which means, in theory, that there's more bacterial growth in the microgravity.
And so the final test we did was look at the bacteria under a microscope. And here we have a picture of what the bacteria looked like after returning from microgravity. When we looked at it under the microscope, we saw that the bacteria was very active. The chains in the bacteria were longer. They clumped together, and you can see there's like a big clump in the middle. That's actually a piece of cotton, but on the cotton there's a lot of clustered Lactobacillus, Acidophilus around it. And then we have the ground truth, and here they weren't very healthy. They looked dead, basically. And the chains were small, and they weren't very clustered together, and they weren't active. And in conclusion, we found that our hypothesis was supported. There is a significant impact of microgravity on the growth rate of Lactobacillus acidophilus. And this implies that astronauts may be at risk to develop dental caries more rapidly if living in microgravity conditions.
The future study will be to repeat this test multiple times in a larger sample. We'll also to stop the growth at a specific time. We would like to thank our sponsors, Community Foundation of North Central Massachusetts, Massachusetts Space Grant Consortium, MIT, Workforce Investment Board, Carolina Biological. We'd also like to thank our instructor, Mrs. DiDiego. Without her, we wouldn't have this wonderful opportunity. Any questions?
Great presentation. Was there a relationship between when you were looking at the optical density and then you were looking at the amount of acid production? Was the ratios pretty similar?
Between
when you were looking at the optical density, the growth versus the amount of acid produced?
Well, what we did see was when we did go back into the lactic acid test, what we used was a binder in the lactic acid so that we could see it under the optical density. And you could definitely see that the proportions were about the same when we did the optical density compared to doing it with the lactic acid test. So it was good to see that they had a common answer to it, I guess, if you can put it that way.
Thank you.